My name is Professor Stephen Hanauer from the Feinberg School of Medicine at Northwestern University, and I am the lead author of the manuscript entitled Safety and Efficacy of BI 695501 versus adalimumab reference product in patients with advanced Crohn's disease, the VOLTAIRE-CD study, a multi-center randomized double-blind phase 3 trial, which was published in the Lancet Gastroenterology and Hepatology. On behalf of my co-authors, I am pleased to present the results from the VOLTAIRE-CD study, which aimed to compare the efficacy and safety of BI 695501 with adalimumab reference product in patients with Crohn's disease. Crohn's disease is a chronic inflammatory condition affecting the digestive tract that can cause abdominal pain, fever, diarrhea, and the passage of blood or mucus. Biologics that target tumor necrosis factor alpha, such as the monoclonal antibodies infliximab and adalimumab, have greatly improved the management of patients with Crohn's disease who are not responding to initial therapy with corticosteroids, thiopurines, or methotrexate. However, these drugs remain expensive. Biosimilars have the potential to increase the number of patients able to benefit from biologics, primarily by decreasing the treatment costs. The additional interchangeable designation allows a biosimilar to be substituted without the intervention of the healthcare professional who prescribed the reference product. BI 695501 is a U.S. FDA-approved interchangeable biosimilar to adalimumab reference product. Bioequivalence between BI 695501 and adalimumab reference product has been previously demonstrated in a study of healthy volunteers, in which the primary endpoints were pharmacokinetic parameters. BI 695501 has also demonstrated similar efficacy, safety, and immunogenicity to adalimumab reference product in patients with rheumatoid arthritis and chronic plaque psoriasis. The VOLTAIRE-CD study aimed to compare the efficacy and safety of BI 695501 to adalimumab reference product in patients with Crohn's disease. VOLTAIRE-CD was a phase 3, multi-center, double-blind, parallel group, active comparator trial, which was conducted at 92 treatment centers across countries in Europe and the USA. Patients were aged 18 to 80 years with moderate to severely active Crohn's disease. This was defined as a Crohn's disease activity index score between 220 and 450, with confirmed endoscopic or radiological evidence of mucosal ulceration for longer than 4 months. Patients were also either naive to anti-TNF therapy or were previously treated with infliximab and had developed secondary resistance. Doses of BI 695501 and adalimumab reference product were 160 milligrams on day 1 and 80 milligrams on day 15, followed by 40 milligrams every 2 weeks by subcutaneous injection. Patients with a decrease from baseline in the Crohn's disease activity index of at least 70 points at week 4 were classified as responders and were eligible to continue receiving study treatment. At week 24, patients in the adalimumab reference product group were scheduled to switch to BI 695501, whereas those in the BI 695501 group received the same product throughout. The primary endpoint was the proportion of patients with a clinical response at week 4. This was defined as a decrease from baseline in the Crohn's Disease Activity Index score of at least 70 points. Secondary efficacy endpoints were the proportion of patients with clinical response and the proportion of patients with clinical remission, which was defined as a Crohn's Disease Activity Index score of less than 150 at week 24. Secondary safety endpoints included the proportion of patients with adverse events, serious adverse events of special interest, and injection site reactions. An exploratory non-inferiority margin of 0.76 for the lower limit of the two-sided 90% confidence interval of the risk ratio was used for the primary analysis. This analysis was conducted in a modified full analysis set of all patients who received at least one dose of study medication and had a baseline and at least one post-baseline Crohn's Disease Activity Index assessment. In the primary endpoint analysis, the proportion of patients with a clinical response at week 4 was 90% in the BI695501 group and 94% in the adalimumab reference product group. In an exploratory analysis, clinical response rates at week 4 remained similar between the groups after model adjustment for covariates. The rates were 88% in the BI695501 group and 93% in the adalimumab reference product group, with a risk ratio of 0.945. The associated 90% confidence interval was 0.870 to 1.028. This exploratory analysis indicated that BI695501 is non-inferior to adalimumab reference product because the lower limit of the 90% confidence interval for the risk ratio was above the pre-specified threshold of 0.76. Data for the two secondary efficacy endpoints were similar between treatment groups. After model adjustment for covariates, the risk ratio for week 24 for clinical response was 1.00. Additionally, the risk ratio for clinical remission was 0.900. The confidence intervals both contained 1 for these analyses, and there were no significant between-group differences. The mean CDAI, Crohn's Disease Activity Index scores in both treatment groups decreased markedly between baseline and week 4, showing further small reductions at weeks 12 and 24. These results are shown in the figure. Overall, reductions versus baseline were similar in the two treatment groups. In the safety analysis set, the safety profiles of BI695501 and adalimumab reference product were similar, as shown by treatment emergent adverse events incidence from baseline to week 24. Results from study weeks 24 to 56 showed that BI695501 was similarly well tolerated in patients receiving BI695501 throughout the study, as in those switching from adalimumab reference product to BI695501. The incidence of anti-adalimumab antibody formation was numerically higher in the BI695501 group than the adalimumab reference product group at baseline, week 4, week 24, and week 48. However, across both treatment groups, the titers were generally low and were not considered clinically meaningful. When we compared the incidence of pharmacokinetically relevant anti-drug antibodies, defined as a titer of greater than equal to 256, the two treatment groups showed a similar incidence, confirming the observed similarity in the efficacy, pharmacokinetics, and safety over 48 weeks. These results show similarly high clinical response rates among patients with Crohn's disease treated with either BI695501 or adalimumab reference product. The exploratory analysis of the Crohn's disease activity index rates at week 4 indicated that BI695501 is non-inferior to adalimumab reference product. Overall safety results were similar between the two treatment groups. Patients switching from adalimumab reference product to BI695501 showed no reduction in efficacy and no increase in the incidence of adverse events. Numerically higher proportions of patients who received BI695501 throughout the study tested positive for anti-drug antibodies compared to patients who received adalimumab reference product followed by BI695501. However, post hoc analyses indicated that a similar proportion of patients in both groups had clinically relevant anti-drug antibodies with a titer greater than equal to 256. The similar efficacies and pharmacokinetics of BI695501 and adalimumab reference product reported here are consistent with earlier comparisons of the two drugs in patients with rheumatoid arthritis and chronic plaque psoriasis. The finding supports the principle of extrapolation, which, for BI695501, enabled its approval across 7 adalimumab reference product indications, including Crohn's disease.